Next up, we have Dr. Mirko Corselli from BD Biosciences. The talk title is Resolving Human Circulating Innate Lymphoid Cell Heterogeneity Using Advanced Single-Cell Analysis Technologies.
Thank you so much. Thank you, everybody, for attending today, and my particular thank you for the organizing for giving me the opportunity to talk to you today about my presentation. I just want a confirmation that you can hear me okay.
Yes, you're all good.
Perfect. Thank you. So today we're going to talk about how advancement in single-cell analysis technology allow you for a refined and deeper characterization of cells and eventually cell signature discovery and resolution of heterogeneity. And as you can see from my title, we'll start talking about innate lymphoid cells. And I'll explain in a second why, but biologically, these cells have been getting a lot of interest over the past decade or so because of their role in regulating immune response. They belong to the innate immune system. They belong to the same family of NK cells, the most popular component of the family. But these cells are not cytotoxic. They are regulatory. They produce cytokines that regulate the immune system. And for that role, they are considered the counterpart of the adaptive CD4 positive T cells. And they share some features with those that we will discuss in a second. They are broadly defined and classified into three groups: ILC1, ILC2, and ILC3. And this classification, again, mirrors the phenotypic and functional characteristic of the counterparts, the Th1, Th2, and Th17. And you see, for example, that ILC1, in response to stimulation, produces interferon gamma TNF-alpha just like a Th1 cell. One of the reasons why these cells have become more important is also because excessive activation of these cells can have a role in immunopathologies and a number of diseases as well. But the reason why we chose this cell type for demonstrating the power of new single-cell technology is because these cells are highly heterogeneous, and their definition right now is based on a handful of markers. The total ILCs are defined broadly as lineage negative, CD127 positive. There are two surface markers, CD294 and CD117, that are used to classify these three cells. However, in the recent years, with the advancement of, for example, high parameter cytometry, study was done with mass cytometry with Cytov, a seminal paper by Newell group, or by single-cell RNA sequencing. These studies have shown that there are just more than 3 subsets. These cells are highly heterogeneous, and there's a lot of subsets that we still don't know their identity and their role. And while this has been very important, it's also worth noting that up until recently, protein analysis or RNA analysis had to be performed on distinct experiments and not on the same cell. So we could infer a possible integration of the data, but realistically, we couldn't do the experiment on the same exact cell. And that's the reason why the importance of single-cell multiomics that was nominated as a technology of the year last year, it's because now we have the tools to actually interrogate both protein and RNA expression at the single-cell level. Multiomics, of course, expand to a number of modalities. It doesn't have to be protein RNA per se, but it's the concept that we can analyze more than one modality on an individual cell. And for the rest of the talk, we will focus primarily here and exclusively on protein and RNA. So how do we achieve these integration and true integration of protein and RNA information at the single cell level? What I'm going to discuss with you today is about a workflow and a workflow solution that we've used to deeply characterize ILCs and resolve their heterogeneity, and then eventually develop flow cytometry assays that can be used for high throughput analysis. But let's start breaking down the workflow and talking about some of the technology that allow us to do so. So after preparing our sample, in this case, we are simply eliminating red blood cells. We're starting from a total blood cells without the red blood cells. We're going to prepare to perform sorting, and I'll explain in a second why. And just like a conventional sorting protocol and cell preparation, we stain our sample with a cocktail of fluorescent conjugated antibodies that will be used by the sorter to identify the population of interest and purify them. At the same time, at the same step, simultaneously, we're going to stain and co-stain with a cocktail of antibodies. And these antibodies, instead of being conjugated to a fluorochrome, they are conjugated to oligonucleotides, just small pieces of DNA. This will not be detected by the flow cytometer, will not be utilized for the sorting, but their information will stay on the cell. The cell will be decorated with the antibodies. They will be eventually read through sequencing downstream. So we're going to use antibody-based assay for two different readout methodologies. One, the conventional fluorescence-based flow cytometry analysis and sorting, and then downstream, more protein can be interrogated by sequencing the tag, and each tag will be specific for each specificity, that then we can retrospectively look at each marker. The next step of the workflow is the cell sorting, and it is important to discuss that when we prepare to perform downstream single cell RNA sequencing analysis, like the one that we're going to do for RNA expression, sorting upstream becomes extremely critical and important, especially for a rare population like ILCs. These cells are extremely rare, they're very much rare, less than 0.5%. And what does this mean is that if we were to use a bulk population without enrichment, we will need millions of cells to have a representative population of ILCs. But different from flow cytometry, where you can run millions of samples at no added cost, sequencing cost depends on the number of cells. More cells, more sequencing reads, more cost. So it becomes extremely important in this case that we perform a sort. We get rid of all the cells that we don't want, more than 99% of the cells. In this case, we sorted the total lineage negative CD127 positive ILCs, and we sorted just a couple of thousand. And that was enough for us to perform a downstream now single cell capture with our single cell analysis system, the BD Rhapsody. Then we can prepare at this level, we are capturing both RNA and protein information. We can generate libraries out of both information and then go for sequencing. But at this point, we've sequenced only a few thousand of cells, dramatically reducing the cost of sequencing and focusing only on the cells of interest. From there, we will show how we analyze the data, how do we get to signatures, and how do we get to information that we can use eventually to go back to the board, design a flow cytometry panel, and transfer the assay to an analyzer. Just a quick slide on the information on the panel. So we, as I mentioned, we have a cell sorting panel with our lineage. We're sorting at a very high level the total ILC. And at the same time, hello?
I think you're still here, Mirko, but just your screen stopped sharing.
Okay, sorry. Could you keep hearing me?
Yep, we hear you.
Okay.
Okay,
all right.
Back on track.
Okay, thank you. So here we're going to show that simultaneously we stain with the cell sorting panel with the conventional fluorochromes, and then with an AbSig panel. These are our reagents conjugated with oligonucleotides. And this selection was a broad selection. It was a collection of markers from several papers, something that we know is expressed on T cells. But this was more of an approach of screening for a variety of markers and then identify what markers are meaningful to identify subsets of ILCs. You will also know that we replicated some of the antibodies, so the lineage markers. We used them both in a fluorochrome conjugated format and in an ad oligo format. And this was done strategically to eventually eliminate any other contaminants that could have been carried over through ABSIC analysis later on. So how does the data look? So as I mentioned, the three group of ILCs are conventionally defined based on the differential expression of CD294 and CD117. And we can look at ILC1, ILC2, and ILC3. Now, these data look like flow cytometry data, feel like flow cytometry data, but they're actually ABSIC data. These are sequencing data read through the SIGIC software that was built based on FlowJo structure and architecture. So it looks like a flow cytometry, but we're reading protein expression based on sequencing, non-fluorescence. And we get an exceptional resolution, very much like you will get by flow cytometry. This also brings another critical point that by virtue of being able now to separate these cell types by ABSIC, we were able to sort at a very high level, just one tube of total ILCs, and then further dissect by analysis. Otherwise, we will have to sort three tubes, one for each population, and then perform RNA analysis. But we're taking this two-step approach, very high-level sorting, and then we can drill down by ABSIC analysis. And now I have my three subsets. I can perform dimensionality reduction, just like Disney. We can see some clustering. We can start assessing broadly some of the markers and how they're distributed across the different subsets. And this is Disney, it's very useful for reducing complexity, for visualizing data at the very high level. So what does this analysis tells me is that some markers like CD4 are expressed only in the ILC1. Some markers like CD2 are expressed in both ILC1 and ILC3, or some markers like CD103 are expressed only in a specific subcluster of ILC2. So these data at the very high level confirm that there is heterogeneity between the three classes of ILCs, but also within a conventional group. And so we've performed, so now we have an experiment that we have all these data, and we can really go deep into all these different routes and avenues. But for the interest of time and for this presentation, I will just focus on our deep analysis into ILC1. So we are now gauging on ILC1 based on conventional phenotyping, and we are going to use a targeted gene expression panel of 399 genes and the 40 plus plex of protein to really dig deeper into this group and identify differences in subclusters. We know from the literature that a paper from Ziegler in the past defined that ILC1 already contained three major subgroups based on CD4, CD8, or double negative. And these cells are not present in ILC2 and ILC3. So the first step, this was done by flow cytometry. Again, we can perform gating analysis just like flow using these ABSIC data and really recapitulate the same findings as Ziegler, where we can see that only in ILC1 we can discriminate a CD4 positive, a CD8 positive, and a double negative population. But now on top of this, we have the gate. We can perform a differential gene and protein analysis. And through SIGIC, it's just one click, and you can identify what's upregulated in this population over this population or over this population. And that's how we came up eventually with this specific signature that you can see here, very clear. Double negative population have these clusters of markers. The CD4 positive population, very specific upregulation of these markers, and a small cluster for the CD8. And now what you can appreciate is that while we are accustomed to see analysis like this for RNA only, this information is now taking account of both protein, where it's AB, or gene. So we're really assessing both protein and genes that are differentially expressed in these different subsets. So we are digging deeper and deeper. We started from the total ILCs. We identified the 3 subsets. Now we're getting into the ILC1. We dissect even more into 3 subsets. And now we found even more population. And we have a larger amount of markers that define the population. So this is great. It gives us a lot of biological information. How do we use this information? But most importantly, this was a screening experiment where we are assessing a bunch of markers that we don't know if they're expressed or not, but it's only one donor. So can we now utilize this information to design an assay based on this information on a more high throughput assays like flow cytometry? And that's exactly what we did. What we have done, starting from the 42 markers that we screened by ABSIC, through this differential analysis, we narrowed down to a subset of 20-something markers that were meaningful to define difference between ILC subclusters. So we designed now a multicolor flow cytometry panel, a 21-color panel. And as I mentioned, as I will show, we leverage the information from the multiomic experiments to guide us in the panel design process. And I will show that in the next slide. We went back to the same exact donor to make sure that the same findings observed by ABSIC and RNA-seq were recapitulated by flow. And once we had that confirmation, we expanded our analysis now to 4 healthy donors to see how those signatures are established, and now they are confirmed between and across different donors. So this slide is about the panel design. So as a flow cytometrist, we are very well aware that panel design can be extremely complicated, especially when we go to higher dimensionality. And we know that the importance of biology, we need to have information about expression patterns and co-expression and expression level or antigen density to appropriately assign fluorochromes, taking into consideration spillover and brightness. We know, for example, that dim fluorochromes can be matched with high antigen density markers, and we know that spillover and spread does not affect markers that are not co-expressed. So we use the data from the ABSIC to really create a table. And now ABSIC informed us, for example, that CD25 was expressed only on the CD4 positive subset of ILCs. Sorry, on the double negative, apologies, on the double negative population. Or we, for example, can identify the level of expression of the marker is expressed at dim level. So we can really go now and have these guided panel design based on this information on relative antigen density and co-expression patterns that are used to optimize a high parameter flow cytometry panel. And just one comment that I would like to make here is that different from flow cytometry, now when we sequence, all the markers are sequenced at the same depth. So we can really be confident about relative antigen density and relative antigen expression because we don't have the variability of the fluorochrome brightness like in flow cytometry. I also point out that we included a marker, CD63, that came up from the RNA analysis, and we screened RNAs that code for surface markers. So we checked whether these translated into a protein as well that was equally differentially expressed. So the first step was validation. So this is our ABSIC single-cell RNA-seq analysis. We screened the markers that were differentially expressed. This is our gating strategy, beautiful resolution to get to our ILCs 1, 2, and 3. ILC1, we can now look at CD4, CD8, and double negative, color-coded. And we really wanted to see if we recapitulated the same findings as the ABSIC. So we look at histogram here to look at each marker individually into each subset. So I'd like to bring just your attention on CD161 and CD314 NKG2D, which were expressed only on subsets of double negative and CD8 positive by flow. When we look back at the ABSIC sequencing data, we confirm exactly the same exact pattern. So again, this confirms that results obtained through ABSIC and single-cell RNA-seq can be utilized eventually and are relevant for flow cytometry analysis as well. So there is a correlation there. And also the CD63, that this was a gene panel, there was not a protein, it came up as a gene upregulated in double negative, came up as a protein as well. But we know that biologically, we're not going to look at data this way. We have now tools to look at data in a more advanced way. And there is a variety of algorithms and plugins nowadays that can really help you navigate the complexity of your cells. So I'm going to show you just an example of a workflow that we use to dig deeper into this data. And as I mentioned, the second step was performed on 4 healthy donors. So the first step, as you can see from this workflow, was to use t-SNE to create, to reduce that, to perform dimensionality reduction. And then identified a variety of clusters. And this is just within ILC1. You can already appreciate how much diversity is there. Then we use phenograph as an unsupervised algorithm to really identify those clusters based on differential protein analysis. And phenograph identifies 13 clusters that are here overlaid on top of t-SNE, on the t-SNE map. And this is all our donors. Now we can deconvolute our donors, and we can see that there is a difference in the distribution of the cluster across different donors. And notably, for example, I would like to bring your attention on this orange cluster that we can find, interestingly enough, only present on this donor number 2. This is great, again, so we are finding difference across donor, but overall, at this point, you're left with the question, okay, we have a cluster that is expressed only on donor 2. What is it about this cluster? What's the phenotype of this cluster? And you can go manually and check each marker individually, but again, you will miss the interplay between the different markers. And that's what I would like to introduce and show you the other plugin and algorithm. It's called cluster profiling. So in a sequential way, we do the t-SNE, we do the phenograph, we identify the clusters, and now we use cluster profiling that recognizes all the clusters, showcases all the markers that we have, either as a line here or as a heat map. And if we know from the previous analysis that cluster 3 was the cluster that was uniquely expressed on donor number 2. So we use this information now to ask cluster profiling, what was the expression profile of cluster 3? And now we can assess that these cells are CD4 positive, CD62L high, negative for 45 array, for example, and positive for CD25. So we can start tracing the phenotype of this cluster that we have identified in a completely unsupervised fashion. And so we go back now to our conventional flow cytometry gating strategy, and we create a gating strategy to confirm indeed, based on CD4, CD62L high, 45 array negative, CD25 positive, that this cell indeed was only present in donor number 2. So we can create now a template for analysis, and we have something that we can screen now hundreds of donors if we want, disease, no disease, or different tissues, and see how this population varies. So the last minute of the presentation, I really want to summarize what are defined as a complete work solution that BD provides for an extensive, comprehensive, a very deep analysis of cells of interest, starting from the cell sorting to focus on only cells of interest, the BD Rhapsody solution for single cell capturing. We have target panel as well as whole transcriptome analysis now, depending on your experimental design. This is done through Illumina sequencing, but then we also have the software for analysis, how these information were eventually used for panel design, guide us through the optimal design of a panel based on biological information, and optimization of this panel on a FACS Symphony A5 cell analyzer to go beyond the 18 parameters. The last piece of the puzzle, of course, would be the next question. There's always a next question, right? So you find the phenotype. The next question now would be, okay, what is it about these cluster of cells? What's their function? Are they functionally relevant? And in order to do so, we'll have to go back, sort these cells, and look, for example, at cytokine input or how they interact with other cells and perform cell function analysis. And now we also have the opportunity to do so through the launch, just a recent launch of FACS Symphony S6 cell sorter, which has the same high parameter capability of A5 analyzer. So you can easily transfer the same 21 color panel on the analyzer into a high parameter sorter to go down to that cell of interest through a refined characterization and perform your functional assays of interest. With that, I would like to acknowledge the team. Data were generated by the Applications Immunology team in the Scientific Affairs Department at BD, as well as the single cell multiomic application team. And of course, a team effort here with Floyd as well for the data analysis. I would like to thank you all for the attention as well. If you want to learn more about the biology, and this was just a part of the analysis that we performed, we have a full-length webinar as well as a white paper. You can feel free to reach out to me directly. That's my email information, or to your sales rep of your local sales representative. And with that, I would like to take any question. Thank you so much.
Thank you so much, Mirko. I really appreciate that wonderful talk. That was great. We do have time. We have about 3 minutes left for a couple of questions. So Rohit is asking, could you please provide details on SeqGeek software? Can we plug in data from other genetic sequencing platforms?
From GenSeq platform, I don't know if you can elaborate a little bit more. Okay. But basically, I don't know if it's about sequencing platform other than Illumina, I suppose.
I believe so.
Yes. Yes. So I'll definitely follow up on that and contact with the SeqGeek team. But if you have any file that comes from a sequencer, we go usually through a pipeline that would convert that into a CSV file, and that can, so any CSV file that is generated can be imported into SeqGeek. But I will make sure to reach out if there's any sequencing platform that may not be compatible. I'm not aware, but I'll follow up with the SeqGeek team to know if there's any such issue.
Perfect. Okay, and then I have a quick question for you, or one quick question for you. So when talking about doing the sorting and then looking at the AbSeq and everything, have there been any cross comparisons to see if there's any up or down regulation of certain proteins as a result of cell sorting? So have there been any studies done to see whether or not certain proteins are up or down regulated as a result of going through the sorting process?
That's a great question. There is a lot of concern, of course, of the effect of cell sorting on protein or RNA expression. We have not seen anything like that. Also, worth mentioning that the AbSeq stain, we usually perform that prior to sort. So that will not capture any protein regulation post-sort. So because the antibody will be at the point bound to the cell prior to the sort. We have tested some, we have done some post-sort staining, but we have not observed, as far as I can tell, any significant down or up regulation as a function of cell sorting. That's an excellent question, but I've not seen it.
Okay, great. And I actually did get a message that SeatGeek is platform agnostic. So thank you to Dan McDonald for clarifying on that. So we'll just know that it is platform agnostic with SeatGeek. And that's perfect timing since those are the questions we have and all the time that we have for today. So thank you again, Mirko, for such a wonderful presentation.
Thank you. Thank you, everybody, for your attention.